Our next speaker is Jeanne Loring. She is a professor at the Scripps Research Institute and in the board of the directors of the Summit for Stem Cell Foundation. She's a renowned stem cell scientist driving research on stem cells, genomics, epigenetics, and cell replacement therapy for Parkinson's disease. Please make the stage yours.
Okay, thank you. I want to thank Aubrey for inviting me to this conference. He's been inviting me to conferences, I just realized, for 9 years. And sometimes I show up. So this one is particularly interesting and very timely for me because it's a particular time in the development of my project that I couldn't have explained to you or talked to you about 2 weeks ago. What I'm going to talk to you about is one story about coming up with a method for replacing the dopamine neurons that die in Parkinson's disease. And if I have time, I have 2 more stem cell stories that I think might interest some of you, or at least it'll be a little bit more like Joe Betcroy's entertaining talk. I'll have a little bit of that at the end. Okay, my disclosures: I'm founder and stockholder, and I continue to advise Aspen Neuroscience in San Diego. Aspen has a clinical trial for autologous stem cell therapy for Parkinson's disease. I'm also a scientific advisor to a number of stem cell companies, and I am an ethics advisor to Merck in Germany. I have a lot of interests. And one of my interests is because Sam Altman invested in my company. I was introduced to him by Aubrey, is ChatGPT and its amazing ability to write haikus. So I'd like to introduce my talks now with a haiku written by ChatGPT. This one is about 25 years of human pluripotent stem cell research. And I'll just read it to you. Unlocking life's code, 25 years of promise, stem cells light the path. Now, pluripotent stem cells are not like the other stem cells that might have been mentioned during this meeting. They exist only in cell culture. They aren't in our bodies. They're the only non-cancer cells that continue to proliferate endlessly. And that turns out to be important. They are also able to differentiate into any cell type in the body. And that also turns out to be important. So the 2 sources of pluripotent stem cells are embryos. Those were first made about exactly 25 years ago. Human embryonic stem cells were made from discarded blastocysts from in vitro fertilization clinics. And some of those that were made at that time point were mine. So I've been in this business for a very long time. The other source, which has been a lot more useful for us, is the induced pluripotent stem cells, which were first made in 2006 from mice and then 2007 from humans by Shinya Yamanaka and his group. Shinya won the Nobel Prize for that work in 2012, and I think it's greatly deserved. There are a number of stem cell therapies in clinical development now, so I'm not alone. There are a couple of them for age-related macular degeneration and retinopathies using retinal pigment epithelium. There's one going on. It's been going on for a couple of years at NIH, and also just started. There was one recently started by Regenerative Patch Technologies in California. Parkinson's disease, epilepsy, and leukodystrophies have been treated by neurons and glia. Among the companies that are working on this are Aspen, Blue Rock, Neuron, Novoglia, and Sana. So there is a lot of interest in the nervous system. There is a company studying hair loss using epidermal cells derived from pluripotent stem cells called Stemson. And cancers, natural killer cells, graft versus host and inflammation. You can make mesenchymal stem cells from pluripotent stem cells. There are some advantages to that, a lot of advantages to that, including the ability to characterize them really well. So they aren't primary coming from a person. Heart disorders, Blue Rock HeartWorks and StemCardia. HeartWorks already has an IND for inherited cardiac, or for cardiac defects that occur at birth. And type 1 diabetes. Vertex has already been doing therapies using islet cells derived from pluripotent stem cells. So let's turn to Parkinson's disease. Oh, I want to remind you, you did have pluripotent stem cells at one point in your life, so don't really feel bad about this. They exist when you were a 5-day-old embryo. You had your own pluripotent stem cells. You can have them again, of course, if you reprogram your skin cells to pluripotent stem cells. Parkinson's disease is caused by the loss of dopamine neurons in a particular part of the brain called the substantia nigra. And it results in movement problems, and it really is due to the lack of dopamine delivery to neurons in the brain. By the time the disease is diagnosed, more than half of the dopamine neurons in the substantia nigra, which you can see that little tiny thing with the arrow pointing to it, more than 50% of those have died. The therapy currently, and for a long time now, that has been used for treating Parkinson's disease, is to give people L-DOPA, which is a precursor for dopamine. And it can enhance the ability of the remaining dopamine neurons to make more dopamine. But there is no therapy that reverses or stops the progressive degeneration of dopamine neurons. This is not a really new concept. Back in the 1980s and through the early 2000s, there were a number of studies done using cell replacement therapy, but instead of stem cells, which had not been made yet, fetal tissues were used. And the interesting thing about it is that most of the time, even though it didn't work, these were 6 to 10-week-old fetuses, so they were aborted fetuses, which would make it very challenging to repeat this experiment in the US, at least now. But the interesting thing was most of the time it didn't work, but occasionally it worked beautifully. And that gave all of us an incentive and a belief that if you could do this right, then you could, in fact, you could actually replace the neurons in a Parkinson's disease brain, and the effects could last for 20 years or for the rest of the person's life. I want to point out Kurt Fried there. This is a wonderful book he wrote about his personal experience doing one of these clinical trials in Colorado, healing the brain. And I'm not the only one doing it. In 2017, there was a meeting in Kyoto of a group called GForce, which invited the 4 major players in this field to all come together and share their information about what it was they were planning to do. So I'll just give you a snapshot of this. This is a published paper. And I want to point out just a couple of things. One is that the work that's being done right now in Sweden, and actually it's being done in Sweden, these transplants have started in Sweden. The ones that are going on in New York by Blue Rock, and the ones that have started in Japan by Jun Takahashi in Kyoto, are all using unmatched cells. The first 2 are using embryonic stem cells, which are not matched to any living person. And the third is using a cell line, an iPS cell line, which is HLA matched to some of the population, but not all the population. So all of those 3 approaches require immunosuppression of the people in order for them to not lose those cells. And they're all planning at least a year of immunosuppression, if not more than that. We started out from the very beginning. We wanted to use autologous cells because you can make these cells from anybody. And we have the advantage of not requiring any immunosuppression in people who are already kind of debilitated. Now, we have heard that in the in studies that require immunosuppression, whatever they are, stem cell therapies, one of the problems is loss of the patients because immunosuppression is very hard on them. So there's a benefit to not having to immunosuppress them. There's a good article actually in the Wall Street Journal in March, which I recommend because it explains what we're doing at Aspen Neuroscience. We take a skin biopsy, we grow fibroblasts from that biopsy, we reprogram them using Sendai virus, which does not integrate into induced pluripotent stem cells. We then take those cells and turn them into neurons using a really robust differentiation method. One of the easiest things to make from pluripotent stem cells are neurons. And to make dopamine neurons just takes a couple of tweaks. It's very reliable. So why should we use autologous cells? I mean, I told you one of them was the issue of rejection. It would be better not to have to immunosuppress people. Manufacturing is a different challenge, though, because if you're using one cell line, you can actually use bioreactors to grow the cell line up. If you're doing an autologous therapy, then you have to make the cells from each individual patient. There are different challenges, but they aren't any greater than the challenges of an allogeneic therapy. And one of the challenges, which I'll point out in a minute, of an allogeneic therapy is the high probability of mutations being acquired by the cells as they are scaled up. And since we do not need to scale up the cells, in fact, we only need a few million cells for a treatment, we don't need bioreactors, we can use culture dishes. And interestingly, if we ever do have to redose people, if these are their own cells, they will be frozen and they can get redosed if the cells don't take the first time. They won't be rejected. I want to talk a little bit about the perils of unlimited cell proliferation. I told you iPS cells or pluripotent stem cells do not stop dividing. They will divide forever. And one of the things that happens when a cell divides forever, like in a cancer, is that it acquires genetic aberrations, genomic aberrations. We've known for more than a decade that these cells are notoriously bad at keeping genomic integrity over multiple cell passages. And for other cell lines, that may not matter, but for these, it really does matter. This is from a paper my lab published almost 12 years ago, in which we used SNP genotyping to study a very large number, nearly 200 different pluripotent stem cell lines. And we discovered a lot of duplications, even some of whole chromosomes and certainly small regions of chromosomes. And the reason that they acquire mutations is because there's an evolutionary process going on in the incubator. And what that means, every time a cell divides, it's been estimated that there are 5 to 7 or so mistakes made in the sequence, in regenerating the DNA. And that's fine because we have a lot of space in our genomes for mutations to occur. But it turns out that if you do this too long, that there'll start to be selection in the culture dishes. And there's one particular gene that is very highly selected for in pluripotent stem cells, and that's P53 mutations. Those are the same mutations that you find in about half of human cancers. So not a good thing to have in your cells, but a few years ago, one of my colleagues at Harvard, Kevin Egan, discovered that many cell banks, even GMP cell banks of embryonic stem cells, contained subpopulations, some large subpopulations, that had P53 mutations. So if you don't monitor your cells and you grow up a lot of them, you have to throw out the whole lot. The advantage of growing up just a few cells is that we have a fail-fast strategy. I'll show you in a moment. So the idea is for us to monitor the genome of these cells frequently over time. So when we first make the cells, we do a whole genome sequencing, and we also do SNP genotyping. We use SNP genotyping to identify the person from which those cells came and also to look for structural variations in the genome. And then we do RNA-seq to phenotype the cells, to know what they have become or what they are right now. And we do whole genome sequencing again when we've selected a colony that we're going to use for and grown it up slightly to use for making the dopamine neurons. And then we do whole genome sequence and SNP genotyping once more before those cells go into patients. That's the advantage of whole genome sequencing becoming routine and very easy to do, especially in a place like San Diego, which is where Illumina is. Okay, so here's an early proof of concept study that we did. Because one of the things that people were worried about was that the cells might not act the same way from different individuals. And this is a demonstration that at least 2 cell lines act almost exactly the same. This is the rat model that people use for detecting the ability of the cells to re-innervate in the rat model. You kill off all the dopamine neurons in one side of the brain, and then in that side of the brain, you put your human cells. It's immunosuppressed, of course, so they won't reject them. You'll see that once the rat is lesioned, it starts circling in the direction of the lesion when you give it amphetamine, because that causes massive dopamine release. So a very straightforward, easy to observe animal test. So if the cells are taking hold and they are re-innervating the brain, then the rats will stop circling in a particular direction. They'll start circling equally. And you can see in the lower right-hand corner, that's one of our rats that's been cured of the lesion we gave it. And the graph shows that these 2 cell lines that we looked at had almost exactly the same timing of recovery in the rats. So I told you we were doing RNA sequencing. The purpose of RNA sequencing is to define the phenotype, but also to predict what those cells can do. So the first of these was an assay called Pluripotent, which has been very popular. It's become one of the major assays to show that your cells are pluripotent, that is, they can make all 3 germ layers. And we did that by setting up a bank of cell lines. We looked at hundreds and hundreds of cell lines that had all been proven to be able to make all 3 germ lines by an assay which is incredibly cumbersome called the teratoma assay. So once we published the Pluritest, people did not use that teratoma assay anymore. So you'll see that I've shown the pluripotency. There's another stage that is called a specified stage, and this is in differentiation of cells. Then there's a determined stage. At the determined stage, they can only do one thing, but they're not that cell type yet. They've limited their options, but they aren't differentiated. And that turns out to be really important because we needed a way of determining whether we had the right cells or not. So we took a lesson from Pluritest and we started doing gene expression profiling. We set up a database and we came up with a predictive assay for cells that would be at the right stage for maximum ability to recover in the brain. And we just published this work last month, some of it, and what I wanted to point out is there's a sweet spot in the differentiation, and we can define that by gene expression signature. If you keep the cells too long, as we did in this study, we kept them a little bit too long, they couldn't make neurites and cause recovery in the rats. But if they were at the perfect place, they did that really well. I'll show you another example here. So to define the optimal stage for transplantation, we had to define the optimal stage of differentiation. And there are no markers for a determined state. It's something that is predicting what's going to happen in the future. It's not a thing in itself. So what we discovered was that if we had the cells in this effective range, they would grow neurites after they were transplanted, which makes sense, and they recovered, the rats recovered. The cells that were a little bit past that stage also engrafted really well. There was no difference in the grafts. There were just as many dopamine neurons at the end of the experiment in both of those time points, but they wouldn't make neurites. They wouldn't respond to the environment in which they've been transplanted and make synapses with the host. So we have narrowed it down to some really interesting candidates that may be responsible for that. One of them is this transcriptional repressor called REST, which seems to be very ubiquitous in development. And we found that there were, in this example, we found that there were 35 genes that are associated, that are repressed by the RES transcription factor, that are not detectable at the effective stage and become detectable at the ineffective stage. In other words, the neurons have to be, or the neurons to be, have to be poised. They have to be holding off on growing neurites until they actually get into the brain. I know. I'm going to go over just a tiny bit. Sorry. Okay, so progress in trials. There are already 3, as I mentioned earlier: Kyoto, Lund University in Sweden, and Blue Rock Therapeutics have all begun their allogeneic trials. And the big news for me is that we have been approved for our first phase 1 trial, a phase 1/2B trial. We were approved about 10 days ago. So we'll be so we'll be starting that trial before the end of the year, and we'll find out. A clinical trial is an experiment you do in humans. Okay, so here's the announcement. You don't need to know that, but I do want to acknowledge something that was really important. This was a grassroots, grounded effort. The first funding I got for this project came from a group of people with Parkinson's disease and their advocates who climbed Mount Kilimanjaro in 2011. They raised $200,000, which is what I said I needed to start the project. And then they went on to climb up to Everest Base Camp and raise more money. And in fact, this foundation, which was founded for that purpose, raised about $3 million over the next few years, which sustained us. We had grants as well, but it sustained us. And then finally, Aspen, once we founded Aspen Neuroscience 2018, in the next 2 years, we raised $220 million, which is what's going to allow us to do a clinical trial. And these are the people in my lab that moved to Aspen. This was a secret to this jumpstart, was that we started with people who were already in my lab. I left Scripps to become an emeritus, and I brought my lab with me when I started Aspen. And we have support from, I want to mention support from Summit for Stem Cell, but CIRM, NIH, Silverstein Foundation, Jonathan Silverstein is from OrbiMed, and obviously other investors. So here's my haiku for the future of stem cells. Stem cells hold the key to healing and longevity. A bright future lies ahead. That's what ChatGPT tells us. Really quickly, here's the entertaining part. I was not just working on, well, I am still working on Parkinson's disease, but I'm also doing work in space. And this is a haiku for stem cells in space. Cosmic voyage starts, stem cells defy gravity, life blooms in space embrace. I've had 4 missions to the International Space Station, and I'll show you what we sent up and what the results are from our first couple of missions. So one of the things you have to think about when you send stem cells into space is to try to define what question you're going to ask, because who knows what's going to happen. We defined a project that would be to understand the effect of microgravity on the brain, because you can't study people's brains in space. You can take blood samples, you can take biopsies, but you don't take biopsies of the brain, so you can't really tell what's going on. And then this cartoon is one of my favorites. You can be anything you want to be when you grow up, and in this case, the stem cells wanted to be an astronaut. What we did was to make organoids. We took the same stage that we're using to transplant to Parkinson's disease, dopamine neurons, from 2 people, and we wrapped them up along with microglia, which are the immune cells of the brain, to make sort of like a brain-like structure, a simplified only part of the brain that's affected in Parkinson's disease. And we made organoids out of them. And then the secret to our success was we put 1 organoid in 1 cryovial in 1 ml of medium, and we sent them to space for 30 days. We had an exact replicate on the ground, and so we were looking for differences between microgravity and ground. And this is the fun part, because we made the microglia, but then we had to get them to the Cape Canaveral. We had to get them to the Falcon 9 rocket that was going to take them up to the space station, the Dragon capsule, and we got to watch all this. So it docked at the ISS. They installed it essentially because it was autonomous. They didn't require anything else. They just stuck it in the incubator. And then it splashed down, and then we got them. And what happened when we got them back, we discovered that they grew like crazy if we put them in a culture dish and let them adhere to something, they grew neurites like crazy when they came back from space. So it was obvious that space had not hurt them in any way. It hadn't killed them. It hadn't suppressed them from doing anything in particular. And we actually, by our gene expression analysis, and the paper, it was just posted in bioRxiv, so I won't tell you all the details. But we discovered we had like 100 different samples, because each organoid was in a separate tube. So we did RNA-seq on each one of those organoids, and we discovered that consistently the cells were more mature when they were in space than the same cells on the ground, by a lot. And don't ask me what my hypothesis is, because so far my hypothesis is really crazy, and I have to form it a little bit better before I go on. And as I said, the most fun part was watching the takeoff. This was the flight that we had last year. And in this case, I actually sent my own iPS cells up to the space station. We made them into dopamine neurons, mixed them with my microglia made from the same cells, and sent them into space. So that was the closest I would probably ever be to being an astronaut myself, but it really felt good to have something of mine up there. This is a collaboration I started with the San Diego Zoo, which is a fabulous zoo, and they have a conservation center there that has 10,000 frozen specimens in liquid nitrogen tanks. This is called the Frozen Zoo. And a lot of those specimens happen to be fibroblasts, which were the same kind of cells that we reprogram to make the dopamine neurons for Parkinson's disease. And they're not that far apart. It takes me about half an hour to drive there, and it's a really fun place to work. So in 2011, it took us a few years, we made the first induced pluripotent stem cells from any endangered species. And the animals that we chose was one called a drill, which is a monkey primate. Not surprising that all we had were the human Yamanaka factors, but they worked. But very surprising, the other animal that we could reprogram is the northern white rhino with the human factors, and they worked. The northern white rhino only has 2 individuals left. It is functionally extinct. 2 females can't reproduce. And this is the cover of Nature Methods that, it's very optimistically suggests that we can bring back all sorts of endangered species using stem cells. So that's what we're trying to do. We came up with a plan with a lot of collaborators to try to make sperm and eggs from these induced pluripotent stem cells. We now have 9 different iPS cell lines from 9 different individuals, which is a lot more than are alive right now. And we're following in the footsteps of the mouse, which everybody does, because they've been very successful in making oocytes from pluripotent stem cells from mouse. And in fact, they can make mice from them. So we're hoping that that kind of work, and also the work that's going on at companies like Conception in the Bay Area, in making human oocytes, that we'll be able to figure out how to do this. And luckily, the zoo has already found our surrogate mothers that will incubate the pregnancies. There are 9 southern white rhinos. They're very similar. And they are now giving birth to babies to show that they can get pregnant. But they are planning to use them as the surrogates for the embryos that we can make with our iPS cells. So if you're interested in this work and you want to donate somewhere, other than to a startup longevity company, or maybe you could figure out why this has something to do with longevity, you can donate to the Rhino Rescue Center at the San Diego Safari Park. And if you go there, they'll let you come and visit the rhinos, like I'm doing there on the lower right. Thanks.